Welcome back everyone. I am saptaparni from BioLive. Hope all of you are doing very well. So today in this video we are going to discuss about metamorphosis in Ascidia. Ascidia comes under phylum Chordata, subphylum Urochordata or Tunicata and class Ascidiaceae. Okay. So in Ascidia we can find all the chordate characteristics in its larval form, and during its metamorphosis into adult those chordate characteristics actually disappears. That is why this organism or this class is very important from your exam point of view, like ugzoology or csirnetlifesciences. Okay, so before beginning I would like to request you to kindly like and share this video if you find this video useful, and don't forget to subscribe to my channel for more such informative videos. So let's begin. So see here, who are ascidians? Ascidians are brightly colored marine animals. In this picture you can clearly see these are adult ascidians who are very brightly colored, pink colored marine animals. Okay, and as I already said, they come under phylum Chordata, subphylum Urochordata and class Ascidiaceae. Okay, some of them are solitary while others are colonial. So both forms are present. And another thing is to remember that adults are sessile, that means they are attached or non-motile. Okay, see, they are adult ascidians are attached to the substratum and they feed. Okay, so they take food, but their larvae, those are Ascidian tadpole larvae, are actually very active and free swimming, planktonic in nature, and they don't feed. Okay, they are non-feeding. Okay, that is the clear difference between the larval form and the adult form. And the most important thing is that most of the chordate characteristics are present at the larval stage, whereas those characteristics disappear during their metamorphosis into adult. Okay, so we are going to deal with this sentence in our next few minutes. Okay, so let's begin. So first of all, let's take a look at the Ascidian tadpole larvae and their characteristics. As we already said that they are non-feeding, planktonic or free living, free swimming, and after their very short time of free living journey, they select a site, mainly a dark shaded site, and they actually attach to the substratum to start their metamorphosis and to become an adult. Okay, larval stage actually exhibits all the chordate characteristics. Okay, so there are three main chordate characteristics, as we all know. First one is presence of pharyngeal gill slit in any stage of life cycle. So in the larval condition of Ascidia we can see small pharynx with slits. See, this one is the Ascidian larva. Okay, and here this part has been zoomed, and here you can see clearly that in its head portion, this is the sorry some yes, this is the head portion. Okay, this is the head portion of the Ascidian larva and this is the tail portion. So we have zoomed this part so that we can clearly see the head or trunk portion here and some part of the tail. So see here in the trunk portion you can see there is a small pharynx. This is the small pharynx with several slits. These are the pharyngeal gill slits. Okay, gill slits, not the gill slits, pharyngeal slits. So this is a very important characteristics for chordate, and in the larval condition of ascidians we can see that. Okay, second important characteristic is presence of tubular dorsal hollow nerve cord. Okay, and third characteristic is presence of notochord. So in chordates we can see that there must be presence of notochord at any stage of life cycle and also the dorsal hollow nerve cord. Okay, so in Ascidian tadpole larvae, in the larval condition we can find both of them, but important thing is to note that both of them are mainly present at the tail portion. Okay, they are mainly present, both the notochord and dorsal holotubular nerve cord are mainly present at the, see here, this part, the tail portion of the tadpole larvae. Okay, so that is why they are also known as urochordates. As we all know, Euro means tail, okay, and here chordate means notochord. So as the notochord is only present at the tail region, so these animals are also called urochordates. Okay. Now going into details of these three basic chordate characteristics. First of all, the notochord. See, notochord is mainly made up of vacuolated notochordal cells, but in case of Ascidian larva, we can see that there is no vacuolated cells. Absence of vacuolated cells are seen in case of Ascidian tadpole larva. Rather, the Ascidian notochord is tubular in nature. Okay, see here, this is a cross section of tail portion. This one is the tail portion, okay, and this cross section is zoomed here. So in this cross section of the tail portion you can find the notochord at the center. This is the notochord, okay, and this notochord is mainly made up of these cells. These are nothing but the epidermal cells, and these epidermal cells actually encircles a central lumen, okay, or a central gap region which is filled with fluid. So obviously this notochord is not solid, rather it is a tubular structure, okay, made up of central lumen encircled by epithelial cells, okay, and outer coating is made up of collagenous sheath. So this is the notochord of our Ascidian larvae. Okay, next going to the gut. See, as we already know that Ascidian larvae are non-feeding, so obviously the gut will not be so much differentiated. As a result, maybe anus is present, but anus is not working until it develops into an adult. Okay, see in this picture, picture of the Ascidian larvae, you can find that this part is the gut part, okay, but this gut is not so well developed, and here you can see this is the anus. This is the anus. See here, it is pointed as anus, but this anus is not working in larval condition. It starts its work during when it metamorphoses into an adult, and a fecal strand comes out from this anus, okay, which indicates that the metamorphosis has completed successfully. Now coming to the post-anal tail. Post-anal tail is one of the major characteristics of chordates. Okay, so in case of Ascidian larva we can also find post-anal tail. See, this is the tail region, so obviously post-anal tail is present, but important thing is to note that this post-anal tail does not contain any myomeres. Okay, myomeres, what are myomeres? Myomeres are V-shaped block of muscles. Rather, these tails contain chains or sheet of muscle cells which are joined by myomuscular and gap junctions. Okay, so this is a important thing to note in case of post-anal tail of Ascidian larva. Another thing is the whole body of these larva is covered by a tunic. Okay, tunic, what is tunic? In our school days we all have tunic. Okay, so tunic is nothing but the external covering. This is the tunic. Okay, so and this tunic is made up of tunisin protein and some cellulose like polysaccharides. Now this tunic actually covers the outer side of the epidermis. So see here, this is the same cross section of the tail region we have already seen. This part is the notochord part, okay, and outside the notochord you can find these are the muscles of tail. Okay, so these are muscles, and outer side of this muscle, this layer, the layer I am marking, this layer is the epidermal layer. Okay, epidermal layer, and outer this epidermal layer there is a presence of tunic. Now this tunic is encircled by two cuticular layer. One is inner cuticular layer and another one is outer cuticular layer. This layer, the most external one is the outer cuticular layer, and this layer, see, this layer is the inner cuticular layer. This layer is the inner cuticular layer. Okay, now you have to remember one thing, that during metamorphosis this outer cuticular layer which forms the dorsal and ventral fin of the tail actually gets absorbed or degenerated. Okay, and after that the inner cuticular layer actually becomes the most outer envelope of the adult Ascidia. Okay, so coming to the next part, the central nervous system of the Ascidian larva. See, the central nervous system of the Ascidian larva is made up of three things. One is sensory vesicle, then visceral ganglion, and obviously the dorsal hollow tubular nerve cord. Now this sensory vesicle and visceral ganglion actually both of them form the rudimentary brain of the larvae. Okay, now let's dig deep into each of them. See, the sensory vesicle actually contains navigational equipment. See, this part is the central nervous system of our Ascidian larva. And if we zoom into this, we can see that the upper portion, this portion is actually the sensory vesicle. Now this sensory vesicle contains two things. One is photosensitive ocellus and another one is gravity sensitive otolith. So obviously this ocellus and otolith together forms the navigational equipment of the CNS. Okay. Next to that, there is a rudimentary cerebral ganglion. This is the cerebral ganglion. Okay, this is the cerebral ganglion, but in larval stage this does not function. This is rudimentary, but when it metamorphoses into an adult it becomes functional. The cerebral ganglion then becomes functional, and that took up the main work of the CNS. Now next to the rudimentary cerebral ganglion there is a visceral ganglion. This larger part is the visceral ganglion. Okay, and visceral ganglion is very important for Ascidian larva because from this ganglion all the nerves are sent to different parts of the body of the larva. Like papillary nerves are sent to the adhesive papilla of the larvae. Okay, then the dorsal hollow tubular nerve cord, this one is the dorsal hollow tubular nerve cord. This one goes to the tail region, and another nerve is caudal sensory nerve, that also goes to the tail region. As we all know, caudal means tail region. Okay, so different nerves are given out from this visceral ganglion. Okay. Next is dorsal hollow nerve cord. Okay, we have already seen that dorsal hollow nerve cord. This one is the dorsal hollow nerve cord, which is present dorsally to the notochord. Okay, in the Ascidian larva, and this dorsal hollow nerve cord actually includes ependymal cells, neurosil, and nerve tracts. How? Let's take a look. See, this is the same cross section of the tail region, and here you can see this red part, this red circle indicates the dorsal hollow tubular nerve cord, which is made up of ependymal cells, and it has a central lumen like notochord, and this central lumen is known as neurosil, and it has some nerve tracts beside it. Okay, so all of these three things make up the dorsal hollow tubular nerve cord. Okay, so this is all about the central nervous system of the Ascidian larva. Next, there are also some new migratory neural crestals which emerges from neural tube, and we have to remember that these kind of migratory neural crestals debut in urochordates, and they ultimately produce pigment cells by differentiation. Okay. Next, coming to the circulatory system, there are circulating blood cells with a rudimentary heart is present. See here, this one is the rudimentary heart. And as I have told about the adhesive papilla, see, these are the adhesive papilla which are now not functional, but these will be the first thing to function during metamorphosis. Okay, these will be functional and these will help to attach the Ascidian larva to the substratum. Okay. So this is all about the Ascidian larval characteristics. Now coming to the main part, the metamorphosis, the journey from Ascidian larva to an adult. So at the end of its short planktonic stage, the Ascidian larva becomes negatively phototactic and positively geotactic. Okay, so then it goes to the dark. Okay, that means it becomes negatively phototactic and it goes to, and it slows down and tries to attach or tries to get into touch with some stone or seaweed, so it becomes positively geotactic. Okay, and they actually find some dark or shaded place, and with the help of adhesive papilla, I have already told you these are the adhesive papilla. Okay, so with the help of these, they attach to the substrate, attach to the substrate, and they starts their metamorphosis into an young adult. Okay, so see, look at this picture. These are the adhesive papilla of our Ascidian tadpole larva. With the help of these adhesive papilla, they attach to some stone or seaweed, okay, a firm place, and then they start their metamorphosis. Okay, so what are the basic steps during metamorphosis? First of all, see this tail part. Okay, this tail part which consists of axial complex. Okay, what is axial complex? Axial complex means the muscle, notochord and nerve cord. So let's go into this part. So this notochord, this nerve cord and this muscle, all of these in the tail region constitute the axial complex, okay, which is covered by inner and outer cuticular layer and obviously the epidermal layer also. So these axial complex along with the tail region, along with the tail, gets degenerated, gets absorbed into the body. See how this part, this tail region is getting absorbed into the body of the Ascidian larva. So first of all, within a few minutes of attachment, contraction of either notochordal or epidermal cells draws the tail. Okay, so the notochordal or epidermal cells gets contracted, which results in drawing inside of the tail along with all its components. What is the component? The axial complex, which is made up of notochord, dorsal holotubular nerve cord and tail muscle inside the body. Okay, so first of all, the tail gets incorporated, absorbed inside the body of the Ascidian larva. Okay, then these notochordal cells separate from each other. First of all, they contract, and obviously when they are contracted, they separate from each other. As a result, the liquid inside the lumen of the notochord gets leaked. Okay, so as a result, the extracellular fluid leaks from the central lumen and the notochord becomes limp. Okay, so as the notochord becomes limp, the axial complex gets resolved and within next several days, its constituent gets redistributed to support the young juvenile adult. Okay, as this is getting absorbed, so their components are now working to make up the young adult. Okay. So along with that, the outer tunic layer, I have already told you there are two tunic layers, outer and inner, and the outer tunic layer also gets degenerated or absorbed during this process along with the sensory vesicles and visceral ganglion. All of these things actually gets reabsorbed. So see here, the notochord, the dorsal hollow nerve cord, the visceral ganglion, sensory vesicles, all the chordate characteristics are getting disappeared during the metamorphosis process. So see here, this is very important to note that. So most of the chordate features that made their debut in the larval stage, namely notochord, tail, post-anal tail, dorsal holotubular nerve cord gets disappeared during formation of adult. Okay, so this is the beauty of Ascidian metamorphosis. Okay, all the chordate characteristics are getting disappeared and they becomes much more like non-chordates, but as their larval conditions retain the chordate characteristics, so we are classifying them as chordates. Okay, so not every chordate characteristics are getting disappeared. Some also gets retained and gets broadened. Okay, what are those? The pharynx. The pharynx enlarges and the slits, pharyngeal slits, increases in number and subdivides repeatedly, producing smaller openings known as stigmata. So another chordate characteristics, the pharyngeal gill slit. This one does not get disappeared, rather it enlarges and this broadens with during the metamorphosis. So okay, that is why we cannot consider the adult Ascidian as a non-chordate, because obviously one of the chordate characteristics is retained, that is the pharyngeal gill slits. Okay, so these remodeled pharynx form a barrel-shaped branchial basket. So see here, in the larval condition, the pharynx was very small with pharyngeal gill slit. As time passed, the pharynx becomes much more broader, and see here how much broader it is. Okay, and now we can call it as branchial basket. And not only its size gets bigger, along with that the number of gill slits also or the number of stigmata also increases. Okay, next the branchial basket rotates to reposition the siphons and the fecal strand appears. See here, if this is the adhesive papilla's position and these are the position of branchial siphon and atrial siphon and the branchial basket itself, see how it repositions, it moves. Okay, it moves almost 90 degrees. See, almost 90 degrees. Now here it is the adhesive papilla, and now their the mouth of the branchial siphon and the mouth of the atrial siphon is repositioned. Okay, and obviously you can see here, at the first I have told you fecal strand is getting out from here, and see here this is the anus, and from anus you can find fecal strand is moving out, and this fecal strand represents that the adult has started feeding. Okay, so the metamorphosis is complete. Okay, see here the fecal strand is present. So attached individual begins feeding, and the appearance of fecal strand testifies that feeding has begun. Okay, so along with the branchial basket, obviously the trunk also becomes broadened. Post-pharyngeal portion of the gut gets regionated into different parts. See here, if this is the pharynx, this is the post-pharyngeal part, and here the gut now differentiates very efficiently to perform all the feeding habits. Okay, and atrium becomes more extensive. Obviously see here the atrium has becomes more extensive, and obviously gonads and gonoducts appear from the mesoderm. Okay, so these are the other things that gets incorporated during metamorphosis. The nerve cord becomes restricted to the trunk region and ultimately gets reduced into a solid nerve ganglion. See, because all the visceral ganglions and other parts of the CNS that are sensory vesicles are now degenerated. Only cerebral ganglion is present, so only a solid nerve ganglion is remained. Okay, so this is the overall process of metamorphosis from larva to adult condition, and here you can very easily see these are the adult Ascidians. They are releasing ovum and sperm, and zygote is forming, then cleavage is occurring. Okay, cleavage is occurring, and ultimately after gastrulation this larva is formed. Okay, and then this larva attaches to the substratum and starts metamorphosis into an young adult, and again they are becoming adult, and then they will will again release the sperms and ovum, and the cycle will continue. Okay, so this is all about the metamorphosis. Now, after metamorphosis, we must look into the characteristics of adult Ascidia also. Okay, we have look into the characteristics of larval Ascidia, and now we must look into the adult characteristics also. So first characteristic of adult is presence of tunic. Okay, tunic was also present in the larval stage also. Now, tunic is made up of tunicin protein and some polysaccharides similar to plant cellulose. Okay, this tunic encloses the branchial basket and the atrial cavity around the basket, and the viscera that are enclosed within this. See, if this is the tunic, this gray part is the tunic. Okay, so it encloses the branchial basket. This is the branchial basket. This is the branchial basket, and around this branchial basket, this part is the atrium. Okay, so the tunic houses the branchial basket, large branchial basket, and the atrium. Okay, now there are two siphons or two mouths, we can say. One is branchial siphon, which is incurrent in nature, and another one is excurrent atrial siphon. See, this is the incurrent branchial siphon, through which water enters inside the branchial chamber. Okay, and this is the excurrent atrial siphon, through which water exits. Okay, so during entry of water through the incurrent branchial siphon, we can find some sensory oral tentacles. Okay, these are some oral tentacles. What is the function of these oral tentacles? They actually taste the water, and they also prevent the entry of larger particles along with the water. Okay, so there are some tiny finger like sensory tentacles which examine the entering water and also exclude excessively large particles before the water enters into the branchial basket. Now, after entering into the branchial basket, you can see the branchial basket is perforated by several pores or stigmatas. Okay, so now how the water actually carries so many foods. Okay, the Ascidians are macrophagous in nature, so they need some food. So with this water, all their foods are getting incorporated into the branchial basket. Now, how will they extract the food from the water, and then how will they excrete the remnant water through the atrial chamber? Now we will look into that. See, there are two structures. Okay, one is the endostyle. See, this is the endostyle. Endostyle is, which one is endostyle? I am marking it with red. Okay, see here, this one, this tract is known as endostyle. It is a ciliated tract which secretes mucus. See, if we cross section these, if we take a cross section of this part, this is the cross section. Okay, sorry, this is the cross section. You can see here in this cross section, this is the endostyle that is present at the ventral side. This is the ventral side. So this is the endostyle, which is a ciliated tract and which secrete mucus. Okay, so when water entered through this branchial basket, and these are the stigmatas. See here, these perforated ones are the stigmatas carrying rows of cilia. Okay, so when water enter inside this branchial basket, these endostyle secrete some mucus, and the mucus actually helps to trap the food particles from the water, and these row of cilia along the stigmata helps to move those mucus laden food materials to the dorsal side. Okay, to the dorsal side of the branchial basket, where dorsal lamina is present. So obviously, when water enters inside these, water carries some food particles, and those food particles are trapped by endostyle released mucus, and those are actually circulated to the dorsal part. This is the dorsal lamina, okay, which is also a ciliated part, to this dorsal lamina by the movement of rows of cilia along the perforation or the stigmata. Okay, and from there actually the food is trapped and it goes to the gut. See here, this is the esophagus, this is the stomach and intestinal portion, and this is, and then through this the excretory material is given out to the anus, okay, and the excretory material is moved out through atrial siphon, and the excess water is excess water comes out through this pore and goes to the atrial chamber and goes out through atrial siphon. Okay, so let's take a look. See, the complex pharyngeal slits or stigmata sieve the passing water before it flows from the branchial basket into the atrium, and from there the current exits via the excurrent siphon. Okay, the rows of cilia, the rows of cilia line the branchial basket. See, rows of cilia are lining the branchial basket. Okay, and the mucus producing endostyle, a mid-ventral groove, is connected by continuous ciliated bands or tracts around the inside of the dorsal lamina. Particulate matter is extracted from the passing stream of water by a net-like sheet of mucus lining the branchial basket, and the rows of cilia collect the food-laden mucus and to move it from the ventral to dorsal, delivering it ultimately to the dorsal lamina, which in turn conveys it posteriorly to the gut. Okay, so we have seen the feeding style or the feeding pathway of adult Ascidia through this. Okay, and see here, these are nothing but the zoomed in picture of stigmata. See, these are the stigmata. Okay, these are the stigmata. Now, along with that, we can find a tubular heart in our adult Ascidia, made up of single layer of muscle-like striated myoepithelial cells. Okay, contraction of heart pushes blood out to all the organs and also to the tunic, and after few minutes the flow just reverse and blood return to the heart. Okay, we have to remember that Ascidian blood vessel is not lined by any kind of endothelium. Okay, instead they are the true hemocysts. You have to remember this one. Ascidian blood contains phagocytic amoebocytes also. Okay, so these are some important points like for the circulatory system of adult Ascidia. There are no specialized excretory organs are present. Okay, and for nervous system there is cerebral ganglion present along with subneural glands and ciliated funnel which joins the subneural gland to the branchial basket. See here, this portion is the nervous tissue part. Okay, this is the ganglion, that is the cerebral ganglion. This one is the subneural gland. Okay, and this is the ciliated funnel. Okay, which joins the subneural gland to the branchial baskets. Okay, so this is the nervous part of the adult Ascidia. Another thing is to remember that the Ascidians are also known as sea squat. Why? Because when they are threatened or buffeted, especially during exposure to low tides, their body masses, there are several striated muscles along their body. Those body muscles quickly contract and reduces the body size Along with that, water is jetted out via the siphons. Okay, so when they are very scared or they are exposed to low tides, their body muscle contract and the water comes out through atrial and branchial siphon in the form of jet. That is why they are known as sea squats. Okay, remember that. For reproduction, we have to remember that they are hermaphrodite. That means male and female sex, both type of sex organs are present in the same individual. That means they are monoecious, and though they are monoecious, self-fertilization is rare. Why? Because the maturity of sperm and gonad, the time of maturity of sperm and gonad is different. That is why self-fertilization cannot occur. So they release their sperm and eggs in the water, and there the external fertilization occurs, followed by zygote formation and holoblastic cleavage, which is nearly equal cleavage. Okay, and after within 3 days, free living, very active planktonic non-feeding ascidian tadpole larva emerges. Okay, and we have to remember that there are two types of reproductive mode. One is sexual mode by the genesis of sperm and ovum, and another one is asexual mode. In case of colonial ascidians, both sexual and asexual mode of reproduction can be seen, and asexual mode mainly involves the budding process from the stolons. Okay, so these are all about adult characteristics of adult ascidians. So in this video we have covered the larval characteristics, adult characteristics, as well as the metamorphosis from larva into an adult. Okay, and we have to remember one thing, that what is the significance. Significance is that this metamorphosis process, or the presence of chordate characteristics in the larval condition of urochordates or larval condition of ascidians, actually indicates the first instance of first instance of expression of chordate characteristics in the world. Okay, in the animal world. So that is why this is very important, okay, from your evolutionary perspective. So I hope this video is useful for you, especially for the undergraduate zoology student. If you find this video useful, don't forget to hit the like button and share it with your friends, and don't forget to subscribe the channel for more such videos. Thanks for watching and happy learning. One thing is that if you want to note some other important points, I have mostly covered all the important points. Not mostly, I have actually covered all the important points, and the reference book is Kenneth Carden. Okay, I have mainly covered all the points from this book only. Okay, so you can also refer to this book. I hope the video is useful for you. Thanks for watching and happy learning.